Section 8.2, the structure of DNA. Okay, so thinking back, way back to chapter 2, we covered in first semester, we talked about nucleic acids. The two types of DNA, nucleic acids are DNA and RNA. Today we're talking about DNA specifically, what DNA is composed of. DNA is one of the large biological molecules composed of these repeating units, these monomers, called nucleotides. Okay, nucleotides. So the parts of a nucleotide, remember the nucleotide is the monomer of a DNA molecule. It's composed of a phosphate group, right here, covalently bonded to a sugar, which is deoxyribose, and then one of four nitrogen-containing bases, adenine, thymine, guanine, and cytosine, which are the bases found in DNA, adenine, thymine, guanine, cytosine, given by ATGC. Okay, so a nucleotide, that's the monomer of a nucleic acid, has these three parts to it: phosphate, sugar, and one of four different types of bases. Since the basic structure of a nucleotide is all the same, they have a phosphate, sugar, and base. The only thing that makes them different is what type or which one of the four nitrogen-containing bases they have. Okay, so this first one right here, this would be thymine, so the phosphate here, deoxyribose sugar, and thymine would be the nitrogen-containing base. Okay, the phosphate to the sugar is held by a covalent bond. The sugar to the base is held by a covalent bond. Okay, on this side, this would be adenine. Okay, and again, it's got its phosphate covalently bonded to a sugar, covalently bonded to this base, which in this case, this is adenine. Okay, the other two are guanine and cytosine down here. Okay, and again, they have the phosphate sugar base on each side, phosphate sugar base, and phosphate sugar base, and that's what composes nucleotides. That's what makes them different from one another, is what type of nitrogen-containing base they contain. So, what are the four nitrogen bases or nitrogen-containing bases? They fall into two categories: pyrimidines, which would be cytosine and thymine, and the purines, which are adenine and guanine. Okay, now looking at the structure of this, the purines, right? The purines have two rings that compose the base, two carbon-containing rings. Carbon-containing mean they're composed primarily of carbon atoms. Okay, so both adenine and guanine are purines. Okay, the pyrimidines have one ring, one carbon ring, and those are cytosine and thymine. These have one ring. They still have the phosphate, sugar, and base, right? Phosphate, sugar, base, phosphate, sugar, base, phosphate, sugar, base, but the bases are different. The structure of adenine and guanine is different, even though they look similar. The structure of cytosine and thymine is different, even though they look similar. Okay, so that's what makes up the four different nitrogen bases, or four different nucleotides. Back in the early studies of DNA, right before 1950, Erwin Schargraf was studying the DNA of several different organisms, and what he found out was that those same four, adenine, thymine, guanine, and cytosine, those four bases appeared in every living thing that he studied. Now, the amount of each base varied. The amount of adenine was different than the amount of guanine, cytosine, thymine different from cytosine, but these things were all in the same, were all in every living thing. Okay, the DNA of every organism that he studied. Okay, second thing is he noticed that the amount of adenine equaled roughly the amount of thymine, and that the amount of guanine equaled the amount of cytosine roughly. It came up in about the same ratio. This relationship, A being equal to T and G equal to C, became known as Chargaff's rules. This is the rules of base pairing that other scientists used to actually discover the structure of DNA. Okay, so the base that is found in equal amounts to adenine would be thymine, right? Adenine would be equal to thymine. Now, later I'm going to talk about why it happened, but these guys actually bond across the structure of the DNA molecule, but adenine is going to be equal to thymine, okay? One of Chargaff's rules. Which base is found in equal amounts to guanine? Well, the amount of guanine equals the amount of cytosine, just like shown in this drawing here. Guanine equals cytosine. That was the other part of Chargaff's rules, that these bases are actually going to cross. They're actually going to bond across the structure of the DNA molecule and give it its distinctive shape that we're going to talk about in upcoming slides. Okay, guanine equals cytosine. Okay, so James Watson and Francis Crick are probably two of the most noted scientists that studied the structure of DNA. They actually received the Nobel Prize for discovering the structure of DNA, and they figured out that it was the DNA structure is a double helix. Double helix meaning it's like a twisted ladder or a spiral staircase, if you want to think of it like that way. So the bases that cross through the structure, that Chargaff's rules, adenine and thymine, guanine, cytosine would make up like the steps or the rungs of the ladder, and the phosphate and sugar would make up the outside of the ladder. And they were able to make a model of it. You can see in this picture here. Okay, of the structure of DNA. Huge breakthrough in discovering the structure was by Watson and Crick. Rosalind Franklin also contributed to the study of DNA, and specifically the structure of DNA. She was actually taking X-ray photographs of DNA molecules, and the way the pictures came out were how the X-rays bounced off showed that DNA is actually two strands that are regular, consistent width apart, which goes along with Watson and Crick's idea that it's a twisted ladder. Okay, so it looked like this, regular width apart, and the rungs of the ladder be like this. And you got to imagine it's twisted. I'm not much of an artist, but if you were to take this and twist it, that would be the structure of DNA. Okay, but it was her work with X-ray diffraction that actually showed kind of the shape of it. Watson and Crick used her information and Chargaff's information to figure out the actual structure of it. But it was all these scientists collaborating together that was actually able to figure out the structure of DNA. Okay, so a double helix, that's what the structure of DNA is, or two individual strands of DNA that wind around like a twisted ladder. The strands are complementary, meaning they fit together, and are the opposite of each other. Now, this little key here tells you what's going on. Remember, base pairing rules are adenine is going to bond with thymine, and guanine is going to bind with cytosine. Okay, so if you notice on the rungs, adenine is given by the yellow, thymine is given by the green. That's what's making up the rungs. Okay, cytosine blue, guanine red. That's what's making up the rungs. This part of it, this outside rail, would be the phosphate and sugar that makes up the nucleotide. Okay, so all this stuff fits together. Okay, it's said to be complementary because adenine complements thymine, meaning they fit together, they bond together with hydrogen bonds, and guanine complements cytosine. Again, they fit together and they bond together through these hydrogen bonds. Okay, I mentioned this previously, but what type of bond connects the phosphate of one nucleotide to the sugar of another? Okay, so this is the phosphate of one nucleotide and the sugar of the next, and these are connected by covalent bonds. You know, it's a covalent bond because it's given by a solid line. So phosphate to sugar, covalent bond. Phosphate to sugar, covalent bond. Okay, the parts of the nucleotide that make up the sides or the outer rails of the ladder is this sugar phosphate backbone. Okay, so again, it's a phosphate covalently bonded to a sugar, sugar covalently bonded to phosphate, sugar, phosphate. Okay, and then these sugars covalently bond to the appropriate base that makes up the nucleotide. Because remember, each nucleotide is composed of a phosphate, sugar, and base, right? Phosphate, sugar, and base. Phosphate, sugar, and base. And the same thing on this side. Phosphate, sugar, and base. Phosphate, sugar, and base. Phosphate, sugar, and base. Okay? So the sugar phosphate backbone makes up these outer rungs, and the nucleotides make up their rungs, or make up the outer rails, and the nucleotides make up the rungs. Okay, the type of bonds that hold together the double helix, meaning holds the one DNA strand to the other, is hydrogen bonds. Okay, hydrogen bonds are formed between adenine and thymine, and between guanine and cytosine. Now I'm putting the number of hydrogen bonds because that's what happens. Okay, adenine and thymine are held together by 2 hydrogen bonds. Guanine and cytosine held together by 3 hydrogen bonds. But if you're looking at this, okay, this, I'll put a different color. This is one strand of DNA all the way there, and this is the complementary strand of DNA. So what I'm putting the line down, this red ribbon is the phosphate sugar backbone. And at each one of these bases, okay, the thymine on this side bonds with the adenine on this side, and the guanine on this side bonds with the cytosine on this side, all the way down this structure to make this double helix DNA molecule. So what are the base pairing rules? Remember the base pairing rules, or Chargaff's rules, are adenine is going to bond with thymine. Okay, it's going to form 2 hydrogen bonds between adenine and thymine. And guanine is going to bond with cytosine, and they will be held together by 3 hydrogen bonds. Okay, along with this, remember adenine and guanine are purines, and thymine and cytosine are pyrimidines. So the base pairing rules is always in a purine bonding with a pyrimidine, right? Adenine, purine, 2 hydrogen bonds, 2 thymine, pyrimidine. Guanine, purine, 3 hydrogen bonds with cytosine, pyrimidine. Is always going to follow those rules without fail.